activities of VALORISH, One Earth, and PROMISEANG projects. I will say just short sentence per each project, because then all the speakers will present in depth the activities. We have VALORISH, that is a project focusing on cascade valorization of fish waste. Then One Earth is focusing on interconnected bioprocessing of agromarine waste byproducts. And PROMISEANG is a project focusing on producing microbial proteins from marine discards. So we have a common thread that is this project starts from animal byproducts, and the overall objective is to identify routes with biotechnological processes and other technologies to create high-value products, or to give another value to byproducts, to give different economical routes and other possibilities to the discards. Here we have Julio Morales and Jaime Villamuera that are my colleagues as cluster managers of the CIRCULARIS BioCluster. I don't know if you want to say good morning or you want to say a few words.
Good morning.
Good morning.
Good morning. Hello. And yes, so I think we can start with the presentations. No, sorry. First of all, I would like to remind the agenda of today. So I will start sharing my screen. Yes, can you see it?
Yes.
Okay. So the agenda for today is divided in the presentation of the three projects. We start with VALORISH, with Laura, Borja, and Marta. Then we will have One Earth. Okay. Thanks to the presentations of Nora and Ana Laura. And then last one, we'll have a presentation about PROMISEANG, but I won't delay much on the topics because there will be...
Hem d'acabar una mica les idees i transmetre-les bé, si veiem que no va per la direcció on nosaltres esperem.
Okay. Yeah, so the speakers will go in depth in these activities. Just a little note that we won't presenting of course, the overall activities of the project, because they've started in different phases, so they are in different implementation phases. What we want to do during today's webinar is to present the results, but also give a space to the participants, which I really thank for being here. We have researchers, we have partners from the industry as well, to exchange knowledge, but also give a space for questions, for technical questions, or even to identify opportunities and synergies between the projects or the different partners that are part of the consortiums, and also, of course, networking. So I will start with the first presentation from VALORISH, presented by Laura. Laura is a research and development technician in Pescanova, and she has a degree in environmental sciences and marine sciences, and a master's degree in science and technology for the preservation of fisheries products. So she will be presenting the feedstock analysis, selection, and preparation for processing of the VALORISH project. So I will leave the space to Laura.
Thank you, Leticia, for the introduction. For those who don't know Pescanova, as she said, I'm working for this company, and it is a multinational company that is specialized in in fishing, in cultivating, and in transforming or processing fish products and seafood products. And we are involved here in this project, in VALORISH project, as industrial partner, mainly. Okay, so can you move? Thank you. The main goal of the VALORISH project is to develop and scale up a computationally assisted biorefinery for valorizing waste and byproducts from the fish industry. And this will be carried out in different steps. The determination of the analytical composition of feedstocks, the oil extraction by applying emerging technologies, proteolytic hydrolysis via fermentation to obtain FPH, support and assistance from computational tools to select the optimal bacteria for that previous task. With the purified FPH, the team will obtain different bioproducts to use and to validate them in food formulations. Also, residual fish bones will be treated to extract collagen, and residual biomass will be also valorized by anaerobic digestion. Let's move to the next slide. Okay, well, this slide is just to have an overview of the project and understand where we are right now. We are, as you can see here, we are halfway through the second year of the project, and almost all work packages have started or are about to do it in next months. Next, please. Today, we are going to present progress and first results of this first work package mainly, which is feedstocks management, extraction, and hydrolysis biorefinery technologies.
Adiós, Sergio. Estamos hablando por WhatsApp.
Oui.
Chao.
Just a quick reminder to keep your microphones off.
Thank you. No problem. Okay, so in this work package, there are 4 different tasks that interact with other work packages. The first task is the identification and characterization of waste and byproducts. The second task consists of the oil extraction and leads to the work package 3, as you can see here in the diagram. Third task is related to the proteolytic fermentation with support of work package 4, and task 1.4 plans to obtain the hydrolysis for using in work package 2 and 3. So getting into the matter, please, next slide. Okay, first task. In this first task, we identify different byproducts from different and diverse factories of the fishing and the processing industry. As you can see here, we identified up to 10 byproducts to be analyzed. First, whole non-compliant shrimps, already cooked. These are, for example, pieces with dark heads. Mass remains and sawdust from cephalopods after cutting processes. Hay sawdust and little crumbles from cutting fish blocks. Surimi paste, already cooked, from producing surimi crab sticks. Cod byproducts from the manufacturing process of filleting or portions. There are also discards originated in the sardine canning industry, consisting mainly of sardine heads or sardine tails. Tuna canning industry is also represented here with tuna dark muscle or tuna dark meat. Fruit byproducts consisting of heads, bones, and trimmings, blue shark skin, and salmon heads and skin. So in the next slide, we can see, let's say, the next step that was to make a nutritional characterization of all of them, where we analyze the nutritional profile. I'm not going to share all the specific data, but some things that we find out were, first of all, practically all of them have interesting protein content. For sure, they are fish or seafood. So all of them would be potential candidates and nice candidates for proteolytic fermentation in next step. But as not all of them have interesting oil contents, we decide to make, let's say, a kind of pre-classification with the ones with the major interest from the point of view of the oil extraction and the ones that are maybe more interesting from the point of view of the protein recovery. And having said this, we found out some other interesting facts. Salt content, for example. We saw a huge variability depending on various factors. As we are working with byproducts from real factories, there is always a range of variability on suppliers and also variability on processes. So, for example, in the case of the hake sawdust, salt content may vary depending on the raw material. In the same factory, they have two different suppliers. One supplier that process the fish block with fresh water, and another supplier process the fish block with seawater. So that makes an impact in the salt content, and that was reflected in our nutritional analysis. Another example in the case of salt is the cod byproducts as well. We have been working with a factory that had the possibility of producing at the same time salted cod or light salted cod. So at the end of the day, their byproducts are mixed together. And for that reason, we found important variations in salt content as well. Another interesting fact, for example, in the case of surimi byproducts, are carbohydrates. Carbohydrates are not so relevant in most of the analyzed feedstocks, except the surimi. And it's important to highlight this because this surimi byproduct is made of different ingredients. It's for sure fish, but it also has, in the formulation, it also has oil, water, flavor, starches. So the impact of this composed matrix makes that the carbohydrates represent, in this case, a great part of the composition of this byproduct, even exceeding the protein content. So as you can see, there are some particularities for some of the analyzed feedstocks that must be taken into account for the next steps of the project. And regarding volumes also, the most relevant in terms of availability are salmon and surimi, considering data of last year, 2024. Others, such as sardine and shrimps, are also relevant. And then we have, last but not least, hake sawdust, cod, and cephalopods. So in the next slide, we can see that basing in both premises, the nutritional composition and the availability of the feedstocks, the byproducts selected for the oil extraction, which is task 1.2, are salmon heads and trout heads. And for the protein recovery in task 1.3, shrimp and hake were selected mainly for their protein content, and surimi also for its availability mainly. We think that it is interesting to see how surimi byproduct performs in this case. And apart from this, nutritional profiles of cod and cephalopods were really similar to hake, as they consist basically of, let's say, white meat, without any shells or without any viscera. So the Valorish team considers that it might be a good option to explore the possibility of revalorizing them in a jointly way, going for the proteolytic fermentation with 2 or even with 3 of the feedstocks together. And that was all from my side from this first task. Thank you for your attention. I leave you now with Borja, that's going to present the rest of the tasks.
Thank you, Laura. So I will present Borja, who will present from Valorish the fish oil extraction techniques and fish protein fermentative hydrolysis. Borja is a researcher in ANFACO. Sorry, I move on with the agenda. Researcher in ANFACO. He's a biologist specialized in biotechnology with a PhD in environmental chemical engineering from the University of Coruna. So thank you, Borja. Please go ahead.
Okay, thank you, Leticia, for the nice introduction. Hi, everyone. As Leticia said, I'm Borja, researcher from ANFACO CITMA, and I am going to introduce you to my talk, which is entitled From Discard to Value, Unlocking the Hidden Potential of Fish Byproduct. And during this presentation, I would like to highlight on what and how ANFACO is doing our tasks more than the results we were achieving during the Valorish project. So we can make the externals to understand what we are doing. So next slide, please. Okay, thank you. Well, this is the index of the talk. Next slide. Okay, thank you. So first of all, and to those who don't know about ANFACO, I'm going to take a bit of my time to explain what we are. ANFACO CITMA is the Spanish National Association of Fish and Seafood Canning Manufacturers. It was founded in 1904 with the mission to promote the research and technological development in the field of marine and aquaculture resources. We are located in Vigo, in the region of Galicia, in the northwest of Spain. And a crucial event to us is that in 2016, the new building for our technological center was born, the CITMA, with 3 main areas, which are the analytical technology area, the technical assistance area, and the research and development area, which is the one I belong to. And next slide, please. Within the research and development area. Okay, thank you. There are 4 different departments with different fields of expertise. We have expertise in digitalization and industry 4.0, biotechnology and health, marine resources and aquaculture, and also sustainability and circular economy, which is the one I belong to. Next slide. Thank you. So ANFACO, as Laura said, takes part in the Valorising project. And what is our role? Well, ANFACO is the leader of Work Package 1 and tasks 1.2, 1.3, and 1.4. And Work Package 1 aims the characterization and proper management of different feedstocks by products from the fishing industry in this case, and to extract from them high value added products such as fish oil and protein hydrolysates. And more specifically, Work Package 1 aims the analysis of available amount and composition of different byproducts from the fishing industry, proper pretreatment if needed, extraction of the oil fraction in those oily-rich feedstocks through emerging techniques, and finally, hydrolysis of the protein fraction through proteolytic fermentation. Next slide, please. I'm going to share the same picture that my colleague Laura shared before. You here can see in a more visual way what Package 1 consists of. First, in task 1.1, different byproducts, as Laura said, are going to be identified and characterized, and then depending on their composition, the feedstock will be directed towards oil extraction through 3 different techniques, which are supercritical carbon dioxide, subcritical extraction, and natural deep eutectic solvents. And to those protein-rich byproducts, they will be directed towards bacterial fermentation, but also the residue coming from the oil extraction could be directed to proteolytic fermentation as well. And finally, in the last task of the Work Package 1, a downstream process is subjected to purify the protein hydrolysates. And well, next slide. Thank you. So as said, we are using different techniques for oil extractions, which are considered to be more sustainable compared to the conventional solvents. The first one is natural deep eutectic solvents, or NADEs. NADEs are a mixture of natural, safe, and biodegradable compounds such as sugars, amino acids, organic acids, alcohols, choline chloride, etc. And during the mixing, the different compounds form hydrogen bonds among them, resulting into a final solvent with different properties than those of each of the compounds separately. And this allows us to extract high value-added products, and in this case, oil from the fish byproducts. For Valoris, we have tested 8 different mixtures. Well, the second technique uses water as solvent, but in its subcritical state, which means in temperature conditions above its boiling point, but enough pressure to keep it liquid. Like a pressure cooker. So in these conditions, water exhibits reduced polarity and enhances its ability to extract lipids. Again, for Valoris, we were testing the effect of different temperatures and pressures to know the extraction yield. And last but not least, the third technique is the supercritical carbon dioxide, which uses carbon dioxide as solvent above its supercritical point. And in this state, which depends again on temperature and pressure, the carbon dioxide acts like gas and liquid at the same time, making it a great solvent to extract lipids. And again, for Valoris, we test different temperatures and pressures to study the extraction yield. And worth mentioning that at this point of the project, the output in oil extraction is quite good, having yields ranging from 45 to 95% from the raw material. Next slide, please. Thank you. So here I'm going to show you some videos regarding the two of the extraction processes, what we are developing here in our facilities, NADEs and supercritical carbon dioxide. As said, NADES is a mixture of different compounds. So in the first place, what we are doing is mixing the compounds until a NADES is formed, an homogeneous solution is formed. So if you please click the video, play the video. Yeah, well. Yeah, thank you. So in the second video, once the homogeneous solution is made, we mix it with the raw material and keep the extraction at a proper temperature and during a specific time to extract the oil. And after the process, what we are doing is a basic downstream process which consists of centrifugation. And if you see the picture beside the videos, you can see the outer layer, which is the oil fraction. So we only need to recover it. And finally, regarding the supercritical carbon dioxide in the video, thank you. The raw material is inside the reactor vessel that you can see inside the equipment, and the CO2 is pumped through it by the equipment. So the CO2 goes from the bottom to the top of the reactor vessel, and it is collected in the glass vessel that you can see at the end of the video. So the good part of the supercritical carbon dioxide is that it is too specific to extract non-polar compounds that no downstream processes are needed. So we collect directly the oil from the raw material, and the raw material is then defatted, but without any pollutant or toxic compound, so it can be used again in other processes. So next slide, please. Thank you. As you see, the final product here is the oil. And why fish oil is such important? Because of its high content in omega-3, and more specifically in EPA and DHA. So based on its composition, it can be used in different applications of high value, such as the food sector, cosmetic sector, or the pet food sector. And next slide, please. Okay, so fermentation is the second technology ANFACO is using in the Valorising project, and in this case for the valorization of the protein fraction rather than the lipid fraction. So here we take advantage of the natural ability of specific microorganisms to break down proteins into peptides because of its natural metabolisms. And we use this ability in our favor. So to us, peptides are more interesting than bigger protein, not only because of their possible biological activity, such as antimicrobial, anti-hypertensive, antioxidant, or immunomodulatory, but also because of their technological properties, such as antifoam, emulsifying, or gelling, which makes them interesting for a high variety of sectors. So what we are doing with fermentation in the next slide, basically here, thank you very much, is mixing the raw material with a specific bacteria. Ones are from our culture bank, other were selected with the aid of a partner from the project, IDNR. So we mix the raw material with specific bacteria in a sterile environment and to perform the protein hydrolysis. So first of all, in the first video, we can see some bottles. We are performing bottle experiments as a first a starting point to optimize the process. And then the idea, as you can see in the second video, if you play it, please. Thank you, is to scale up the process to 5-liter bioreactors. So once the process is done, a downstream process is needed and follow the fermentation step. And this downstream process includes centrifugation, filtration, and also drying to finally obtain the hydrolysates, which are the pictures that you can see there. It's a fine dust. The second one is not as fine, the other. But yeah, that's the main idea. So thank you very much for your attention.
Thank you, Werham. Thanks a lot. Really interesting what we can do, how we can play puzzle with fish waste. And next one, we have Marta Franco from Edener. I would just like to have a check on the time, because now from the agenda there was supposed to be a break, so we are currently using the break time, and I invite the speakers to please stick to the 10 minutes around presentation, so we can hear properly all the presentations and give the same time to everyone. Let's move on with this presentation from Marta, who's a senior researcher at Edener, and in the project Valorish, she leads the computational screening of proteolytic bacteria. By training, she's a computational biologist with deep expertise in genomic and proteomic data analysis. So thank you, Marta. Please feel free to present.
Hi, everyone. My name is Marta, and I'm part of the Edener team. I'm going to talk about our in silico, how our in silico work can support the experimental task that my colleagues have just presented. So next slide, please. Okay, here is a quick, oh, sorry. The last slide was a quick overview of my presentation. I will start with the context of our work within Valoris, and then I'm going to introduce our computational workflow, who is formed by 3 main steps, and then I will finish with some quick conclusions to not extend so much this presentation. Okay, and here, this slide has some animation, so sorry there is some chaos. As my colleague Laura explained before, the Valoris project aims to develop sustainable biotechnological strategies to valorize fish waste and byproducts. At Edener, we coordinate this project and are technically responsible for developing the computational and modeling tools that support the design of and the optimization of these bioprocesses. In this presentation, I will focus on the first stage of these computational approaches, which is the screening of the proteolytic bacteria. You can pass through the animations, I think, if it's possible. Yes, and the next animation too. Yeah, thanks. And here we use computational biology to identify the most promising microorganisms for the proteolytic fermentation. And so in this work, we develop a computational framework that integrates different levels of analysis. Starting from a defined set of proteins, substrates, and a group of candidate bacteria, we simulate how each strain could digest those proteins based on their enzymatic profiles, and in parallel, we evaluate their metabolic performance. Finally, what we do in this workflow is to predict the bioactivity of the resulting peptides. This helps us to identify the most promising strains for the further validation in the experimental phases of the project. Next, please. Next slide, please. Sorry, Leticia, I cannot see the next slide.
Sorry, Letizia, maybe Marta can share her presentation, her desktop, so we can go quickly.
Yeah. Yeah, because I cannot see the slides moving. Or
I think that we lost Letizia.
Yeah. But I cannot share my own screen because I have not the permissions. Sorry.
Let's just wait a minute for Letizia to come back because she has the control of the presentation.
I'm back. Sorry.
Okay, perfect.
Okay, I think you cannot see it now, right? Okay. Sorry for this. We were left here.
Yes, in the next one, please. Okay, here. Thank you. Sorry for the interruption. Okay, I was saying that we have a computational workflow that helps us to define the best candidates to perform these proteolytic digestions. But before running this simulation, we first define the protein substrates that will serve as input for our analysis. And here we start from a large dataset of 5 byproducts provided by the partners working on feedstock analysis, Pescanova in this case. And from this initial group, we prioritize 3 species based on the results of this feedstock characterization and selected each the representative protein sequences. These protein sequences were mainly structural proteins such as collagen, actin, or myosin, which are abundant in these materials, are relevant for this proteolytic digestion, and we can use it in our them in our protocol. We obtain these sequences from public databases. And also, next slide, please. Thanks. In parallel to these substrate selections, we also define a group of microorganisms to be tested. A total of 81 microorganisms were selected from species included in the qualified presumption of safety list published by the European Food Safety Authority. What this means? Okay, this means that all of them are considered safe and suitable for food-related application. As you can see in the chart, the most represented genera were Lactobacillus and Bacillus, but there are other genera represented with 1 or 2 strains, which still gives a diverse panel of candidates with proven safety profiles. So now that we have defined what are the inputs of our workflow, which are the substrates at the microbial panel, let's take a look on how the full computational work is structured. So next slide, please. Thanks. Our computational workflow integrates 3 main stages, as I have said at the beginning of the presentation, which together allow us to evaluate the potential of each microbial strain that we are evaluating. The first stage focuses on the in silico enzymatic digestion, and here the selected protein substrates are digested computationally using specific enzyme sets that represent the proteolytic profiles of each of these 81 protein organisms. In the second stage, we evaluate the metabolic feasibility of these microorganisms using genome scale metabolic models. I will explain what genome scale metabolic models are later. And this step helps us to identify which strains are not only enzymatically active, but also metabolically viable on their more realistic nutrient conditions. And finally, there is a third stage in which we assess the bioactive potential of the peptide generated in the simulation. Here we use several prediction tools to estimate whether this peptide might show antimicrobial, anti-inflammatory, anti-hypertensive, or antioxidative properties. So altogether, these 3 steps led us to obtain a complete framework to identify the best performing strains, but that are those who combine strong proteolytic capability, metabolic robustness, and also the ability to produce some potential valuable bioactivity, bioactive peptide. So next slide, please. I will explain these 3 phases in more detail. In this phase, we use an existing computational pipeline, which is called FITS. Basically, this tool takes as input the genomes of the microorganisms and the selected protein substrate, and it then simulates the digestion process, substrate by substrate and strain by strain, identifying the proteases encoded in each genome and predicted how they will cut the proteins. As a result, what we obtain? Okay, we obtain a library of digestive peptides for every microorganism and every substrate. To make this pipeline more realistic or more adapted to our needs, we introduce few improvements. The first one is that we added a step to distinguish between extracellular and intracellular proteases, since only extracellular enzymes are expected to act during the fermentation process. And second, we include a length-based filtering, removing peptides shorter than 3 amino acids because they are not relevant for the activity prediction. And we know this because we have made many tests regarding this part. Next slide, please. Here. And this chart, as result of this step, so here you can see the top 50 microbial strains ranked by the number of peptides generated in the simulations. As you can see, there are some genera such as Xanthomonas or Bacillus that stand out as the best candidate, but just in this stage of the workflow. And next slide, please. Okay, and we're going to pass to the second phase of the workflow. Here we evaluate the metabolic feasibility of each strain using genome scale metabolic models. And as promised, genes are computational representation of an organism's metabolism. They describe how genes, enzymes, and biochemical reactions are connected inside the cell in order to produce energy and biomass. In our workflow, we will strain specific genome scale metabolic models, and this allows us to predict how well each bacteria could grow and how efficiently it could consume these amino acids. The graph that you can see in the slide shows the number of amino acids consumed by each strain and the corresponding biomass production. Ideally, we are looking for strains that show a high amino acid consumption together with high biomass production. And finally, let's move to the third part of this workflow. And in this last phase, we take the peptide libraries generated during the enzymatic digestion, the step 1, and analyze them using different bioactivity prediction tools. Each of these tools estimates the likelihood of that peptide to showing some kind of interesting bioactivity. Whenever it's possible, we have used more than one tool per bioactivity since each model is training with different dataset and use different algorithms. So by comparing the results across different tools for a same bioactivity, we can increase the robustness of our prediction. And in this way, we try to reduce the bias that could come from relying on a single resource. And next slide, please. And here you can see an example of the results for this antimicrobial activity inference part. Each color represents a different precious tool, and as you can clearly see, the number of predicted antimicrobial peptides varies a lot between them. And this variability highlights the utility of applying several tools in parallel. Of course, we should remind that these are in silico predictions, and we are at this moment trying to validate this complete workflow with experimental data that comes from ANFACO task and from their work. And next slide. So, in summary, this computational framework helped us to identify promising bacterial strains, optimize the digestion strategies, and guide the experimental validation of this protocol. In this way, we aim to bring the gap between modeling and real fermentation processes. But as I have said, we are working on validate this protocol, but we are obtaining pretty good results. So thank you so much, and sorry for the interruption.
Yeah, I should say sorry for the interruption, was my fault. Your presentation was great. So I cannot complain if you are late because of the interruption. But we can move on directly with the, now we change project. We go with One Earth. That adding to Valorish, it has also the land stream. For example, we have also chicken feathers and cheese whey as byproducts. But and here Noura Raddadi, Associate Professor of Industrial and Environmental Biotechnology at the University of Bologna, will present the valorization of chicken feather and fish processing byproducts. Noura is also the coordinator of One Earth project, and she has a huge experience in other projects involving marine extremophilic bacteria in biodegradation of plastics and organic compounds. I will give the floor to her. Thank you, Noura.
Okay, thank you, Letizia. So good morning to I am going some of the activities dealing with One Earth project. So this is a project that involves 15 partners from different countries, including 4 research and higher education institutions, 2 research organizations, 5 SMEs, 2 large enterprises, and 2 consulting. Next slide, please. The consortium includes these partners having different skills with the aim of developing and assessing innovative bio-based solutions for the valorization of residual biomass origin into nutraceutical bioadhesive, but also fertilizers and products. Specifically, we considered in the project at least the byproducts of 3 different industries. Cheese whey, that is subjected to anaerobic fermentation processes for the production of fatty acids, as well as CO2 rich gas stream. Both will be exploited by the use of bacteria, yeasts, but also microalgae for the production of saturated fatty acids. Both the production and also downstream transformation are the target of our package that will be covered by the talk of Anna Laura in a few minutes. The other 2 industries considered are the poultry industry, in this case with chicken feeder, and the fish industry byproducts such as chicken, fish scale, and fish bones. All these 3 types of substrates are exploited in work package 3 for the production of peptides and fertilizers. Next slide, please. So the work package 3 is structured in 3 tasks. Task 1 dedicated to the raw material supply, including collection, separation, but also characterization of this raw material that is exploited in task 3.2, production of peptides through different routes, about which I am going later, and task 3.3 dedicated to production of fertilizers and specifically biochar to be applied, of course, for agricultural purposes. Both the biochar obtained in task 3.3, but also the peptides that are obtained through protein hydrolysis in task 3.3 exploited as ingredients for obtaining the products in such as nutraceutical, cosmetic, and adhesives from peptides, but also fertilizers from biochar. Of course, dedicated work package 5 is dealing with the evaluation of the sustainability of these different processes through LCA and LCC analysis. Next slide, please. So the 3 different routes that I am going to cover in this left from my talk are the microbial proteolytic route is developed mainly by University of Bologna, Unibo, the enzymatic and the chemical routes that are covered by FH and W from Netherlands. Next slide, please. So with regard to the first route, we started with the selection of microorganisms through preliminary screening on solid media containing skin in order to select bacteria that are able to produce extracellular proteases. Then from 79, we were able to select 25 isolates that have been further analyzed for their ability to go using chicken feather as main substrate and to complete hydrolyze the substrate. And among these 25, 10 has been selected. The hydrolysates obtained from these 10 microorganisms have been subjected to the evaluation of the bioactivities, and indeed at least 5 hydrolysates exhibited activity, 5 exhibited antioxidant activity, and also 5 were able to produce keratinases that are the main enzymes involved in degradation and hydrolysis of keratin, is the main component of chicken feed. Slide, please. Among these 5 isolates, one was selected for further analysis, and on the hydrolysate obtained by this isolate, we also evaluated the cytotoxicity of the hydrolysates, and we observed no cytotoxic effect for skin cells when we used diluted and autoclaved hydrolysate. On the same hydrolysate, we performed also some other ultrafiltration assays in order to separate the keratinases from the antioxidant compounds, and indeed using 10 kilodalton cutoff membrane, we were able to separate the permeate compound while the keratinase mainly recovered in collaboration with FHNW. We performed also further analysis in order to determine the picture size of the in the high state, and able to detect having molecular weight from 400 on this same isolate, we also performed the whole genome sequencing and some proteomics analysis, and indeed it was possible to detect several cases and first attempts for cloning this keratin are being. Next slide, please. The same approach that we applied for a chicken feather has been considered for fish scale and fish bone, and so in this case we performed different assays for evaluating the ability of bacteria from marine but also environment. At least 2 isolates selected, and again we were able to observe antimicrobial activities toward E. coli, Escherichia coli, but also Staphylococcus aureus, as well antioxidant activity that was also stable to autoclaving. The same approach has been also applied for the separation of keratinases from the antimicrobial process. Next slide, please. So going to enzymatic approaches, in this case, a step for solubilizing the keratin was required, and in this case, phosphinose was utilized. After this step, different enzymes have been evaluated for their ability to hydrolyze the keratin, and at least 2 have been selected as the most promising. Also, analytical methods for evaluation of the molecular weight of the peptides obtained have been developed and have been set up. Next slide, please. This is the activity performed, as I was saying, by FH and W. Next slide. Okay. Another activity always performed by colleagues from FH&W dealt with the selection of performing bioinformatics analysis for selecting other keratinases to be expressed and used for hydrolysis of keratin, and also some experiments have been performed for performing immobilization of the enzymes. The third part that you see in this slide, that is dealing with chemical hydrolysis of the fish scale and fish bone, but also chicken feather. In this case, different concentrations and different incubation times have been considered for acidic and alkaline or alkaline hydrolysis of the feather, and the optimal conditions have been chosen and have been developed for obtaining peptides from chicken feather. This is mainly random hydrolysis, chemical hydrolysis, and other activities dealt with. Next slide, please. With performing bioinformatic analysis for the aim of developing bioinformatic tools in order to be able to identify the peptides obtained based on their mass. And indeed, these approaches are available for peptides obtained through enzymatic hydrolysis, but are not developed yet for chemical hydrolysis. So this is an important activity that is being carried out and that is being developed. The next slide, please. So other activities are ongoing and that are aiming at developing approaches for hydrolyzing in a specific way. So in together in order to obtain peptides having specific. So next slide, please. Okay, so summarizing, for microbial roots, we were able to select bacteria from desert and marine environment that are able to produce subscale hydrolysates having antimicrobial as well as activity from chicken feather, but also from fish scale and fish bone. The hydrolysates have high stability to heat treatment, which is very, but also did not exhibit toxicity towards skin cell. It was possible also to perform different tests and to separate the enzymes, so the keratinases from the antioxidant and microbial compounds from keratinases through. With regard to the enzymatic hydrolysis, it was possible to select at least 2 enzymes for peptide hydrolysis through chicken feeder hydrolysis. Also, the analytical approaches for peptide characterization have been set up, and immobilization techniques have been set up for model enzymes such as tryptophan, while the attempts for applying them to the recombinant keratinases are being performed. Regarding the chemical hydrolysis, the parameters have been optimized, and the analytical tools have been also characterized and developed. Further bioinformatic tools to identify the peptides based on their molecular mass, and consequently to understand better the pathway for the degradation of keratin through chemical analysis are being developed. And other activities are also being performed in order to set up the tools for the selective chemical hydrolysis of the chicken feed. Thank you. That was all from my side. Okay. I am available if you have any question. Otherwise, I apologize because I have to leave.
To leave, I will give the floor to any question for this specific presentation. If not, we have at the end of the presentation some Q&A section. Is there any question? In that case, we will share the presentations with the content if you want to reach directly to Nora. Okay, thanks a lot, Nora.
Okay.
So,
bye-bye. Thank you. Bye.
Bye. Have a nice day.
Thank you.
Now we move on with the webinar presentation on polyunsaturated fatty acid reduction and stream transformation done by Anna Laura Eusebie. I saw her join before.
Yeah, I'm here. You can hear me?
Okay, perfect. Yes.
I don't hear you. I don't hear you perfectly, but I don't know if it's my problem or... But we can go.
Okay, important thing is that you, the participant hear you better than me. And introduction to Anna Laura. She's a professor at the University of Le Marche. She has extensive experience in developing and validating advanced technologies and processes for the treatment and valorization of liquid matrices, primarily in the water sector, with a focus on environmental and carbon footprint impact through extensive international research and coordination in more than 80 international. So I will share again the screen. Okay. Yes.
Okay, thank you a lot.
Okay,
Thank you, Letizia, and all the participants. We will go to show some main results related with the activity developed in One Earth in the work package 2. The object of the work package 2 is mainly the production of the polyunsaturated fatty acid using different type of downstream transformation. We are, as Università Politecnica delle Marche, involved in one specific way to obtain this type of PUFA production, and we will show you some preliminary results according to the timeline of the project. Please, next slide. As you probably know, the activity of cheese and milk productive sector, it's a wide, huge productive and industrial section where we have a lot of waste or byproduct production, usually from 7 to 9 liter of whey waste for each kilogram of cheese is produced, and between 1 and 2, 3 liter of milk is wasted starting from 1 liter of milk processed. So at the end, the amount of this type of subproduct or waste could be interesting, also considering the presence of this type of productive sector in several countries. And this type of flow is characterized by an elevated concentration of COD, so of organic substances, and an elevated quantity of lactose that could be valorized in different ways. So the One Earth approach is more focused on the production of polyunsaturated fatty acid, mainly for our activity that I will show you later, related with the seafood final diet production, because in several other options of valorization of this type of waste, the agricultural sector is the one more centrated in the final route of possible valorization. And for this, there are a lot of scientific publication, techno-economical and environmental assessment, and also activity related with not only lab scale, but also pilot or industrial or full-scale realization. In One Earth, the idea is starting to valorize and produce PUFA for diet to be applied to the seafood production, considering that the seafood is one of the, and the aquaculture in general is one of the key points for the next year probably. And in this sector still lacked the necessary experience for full-scale upgrading, and the validation of the effective valorization added value compound by different way are necessary to be more highlighted. So the main objective of our work is this interlinking valorization approach. Please, next slide. So the work package 2 is connected with this type of idea and objective, and is connected with the 4 different tasks. The first task is produce the volatile fatty acid starting from the cheese whey to be after more elaborated and transformed for the production of the polyunsaturated fatty acid. The second task is related with the production of the PUFA, and the third task is with the valorization of the PUFA-rich algae, and also starting with the and using the CO2 rich effluent from the VFA production for the growing of the algae. The fourth task is connected with the enzymatic esterification of the PUFAs, and the last one with the feed production for the aquaculture sector. As you can see, the partner involved are in part company, in part research or university center, in order to obtain also this practical approach and give some upscaling information and idea of possible technical, economical application of all these type of different way to valorize the cheese whey. Next slide, please. As you can see, this is the main graphical abstract of the work package 2, and as I described in the different task, the cheese whey will be anaerobic fermented to produce a flow rich of VFA. The gas stream will be used for the algae photobioreactor for the microbial transformation, the production of a flow of PUFA rich, and the VFA rich flow in the liquid part will be fermented by yeast and bacteria for the production also of PUFA. The PUFA will be used directly for the application into the aquacultural sector, and so for the feed preparation for seafood. And from the other part, the enzymatic transformation by esterification could be produce added value compound to be used for cosmetic and as nutraceutical ingredient. So the activity that I will describe in the next slides are centered into the part of the anaerobic fermentation and the VFA rich, that is this slide in terms of flow scheme. And what we have done into the activity 2.1 is mainly related with testing the best condition to produce the VFA rich effluent, considering one first phase of anaerobic fermentation, another phase of separation that is a key point into when you work with the presence of flow at higher organic compound concentration. So we have studied the best option to separate the liquid rich in VFA and the concentrate flow. The solid part will be sent to the pyrolysis to be valorized in terms of soil amendment and fertilizer, as Nora already presented before. In the first phase, the achinogenic fermentation at the lab scale was evaluated in terms of also optimal testing condition, and I will show you the main results of this, and also the final key finding aspect is the upscaling of this type of flow scheme in a pre-industrial way. Please, next one. Okay, so when we started to work with achinogenic fermentation at the lab scale by using our system in fermentation scale on small quantity, so some liter of test, we tested both batch mode, so working with the natural pH and to identify an optimal HRT of fermentation, and also we tested in semi-batch condition using different pH conditioning phases, and also inoculated some commercial inoculum to evaluate the possible speed up of the fermentation. Also in the preliminary phase, the process and efficiencies was evaluated in terms of higher performances and VFA production to reduce the HRT and SRT, in order to optimize the scale-up into the pre-industrial scale of the pilot system. The separation aspect were studied, evaluating different type of possible separation of the final fermented flow by using not only physical separation by centrifugation or physical separation, conventional physical separation, but also testing flotation or coagulation and flocculation. Next slides, please. Here you can see in the table the main different tested condition that we considered. As inoculum, we use a probiotic bacterium strain, so a conventional one, and we change a pH, FT, SRT to identify the best VFA growing into the trials that we developed. Next slide, please. As a final evidence, here you can see the preliminary results starting from the cheese whey. The IVFA concentration was obtained in the range between 18 and 20 grams per liter, around the pH of 6.5, and the presence of the use of the propionic bacterium show a better VFA yield compared to the control trial. For sure, the working in the semi-batch help a lot the production of the VFA and no need the nitrogen supply to obtain the anaerobic condition during the fermentation. Finally, the best strategy for the VFA rich separation after the anaerobic fermentation will be identified in terms of using centrifugation plus filtration at 0.45 micrometer. Next slide, please. In terms of VFA profile, the presence of the lactic into the cheese whey will be converted completely in mainly butyric acid, and as a second VFA we will have acetic acid, and the third one is the propionic. The butyric is the dominant, and we develop a small control also to try to identify and to create the best condition to produce a specific type of VFA compared the other one. Next slide, please. This type of approach will be used also into the pre-industrial scale. Checking the correlation, statistical correlation between the different experimental tests and growing obtained in terms of VFA yield, and also as a concentration and as compared to the initial COD and COD, organic carbon present into the original cheese whey, the semi-batch mode was considered the best option, and the optimal parameter identified in order of importance to obtain the final optimal concentration of VFA is related to the pH control, first level, SRT, so sludge retention time control between 12 and 20 days, and HRT as last parameter between 6 and 10 days of contact time. Next slide, please. So from here, we design the scale-up, and actually this is the flow scheme and the P&I of the fermentation plant, characterized by a fermentation volume of 500 liter. It will be coupled with the VFA recovery unit with the possibility to separate the flow both with the centrifuge and with the ceramic membrane in order to obtain in a controlled temperature and by using a chiller system a final flow reach of volatile fatty acid to be sent to the partner in Portugal for the production of the PUFAs. This scale-up is actually in the phase of the realization and will be installed into the pilot of our university, and will be the phase of the startup will be in January with the production of the we hope higher amount of VFA flow for the validation of the flow scheme that I've shown now. So hope to see you for the next webinar with the results of the pre-industrial application of the process. Thank you a lot. Here are our details.
So here you have the contacts if you share for the, but also I invite you to stay here until the end for the Q&A session. So now we are done with the One Earth project presentation, and we move on with the PROMISEANG to Celia. And Celia, I hope that I said it correctly. She's a researcher and biologist at the University of Vigo, and in the project PROMISEANG, she's managing the research of bioactive properties in cell models, and by training she has a PhD in basic and research to veterinary sciences, and her area of expertise comprises marine biotoxins toxicity assessment in vivo and in vitro models. So the floor is yours.
Thank you very much. Can you listen to me properly? Okay, sorry.
Yes.
Yes.
So thank you. I will first give you, well, this is the agenda. I will first give you a brief introduction to the PROMISEANG project. Then I will talk a little bit more about the marine byproducts that we work with, along with their nutritional characterization and the food safety and toxicity screening that we perform, to finalize with the future perspectives and conclusions. Next slide, please. Vale. Okay, so the PROMISEANG project is a European project integrated by public institutions as well as by private companies from 5 different countries: Spain, Portugal, Germany, Poland, and Hungary. The broad aim of the project is to find byproducts to revalorize the discards from fish industry as a source for alternative proteins. Within this aim, we can separate it in smaller objectives to understand the requirements for use of marine food waste and byproducts as fermentation substrate, use biomass fermentation to produce sustainable, healthy, protein-rich microbial extracts, confirm the nutritional value, safety, and health benefits of the new food and feed products, determine the sustainability of the new proteins compared to currently available alternatives, and to foster development of the circular economy by recovering bioactive compounds from fermentation side streams. Next slide, please. So here I show you a simplified workflow of the project. So first we counted with 30 very diverse marine byproducts that were provided by fish industry. I will talk about them in a bit. They will be characterized in the sense of their nutritional potential and their food safety concerns, if there were any. And so based on that results, some of these byproducts will be selected to optimize solid state fermentation and liquid state fermentation. And once this process will be done, the nutritional analysis, bioactivity properties in cell models, and the safety assessment will be then evaluated so that we can generate new products and define and better define the applications of these cards, let's say. So I will be focusing on the work package 2, that it's the first part of the project, which entails the nutritional characterization and the food safety evaluation. Next slide, please. Right. Okay, so I have separated or divided the biomass that we work with in 4 different groups. However, most of them were derived from fish, and it entailed different parts such as dorsal fin, skin, tails, viscera, central spine, and heads, majorly from 2 species of tuna that were the skipjack tuna, Catsuwonus pelamis, and yellowfin tuna, Tunus albacares. From each independently, and then from both of them, we work with a mix of these 2 byproducts in form of decanted meat and cooking broth. And moreover, we work with extra 3 samples from turbot, horse market, and blue whiting that were viscera and heads of the latter 2. Next slide, please. Okay, then the second group will be mollusk bivalves, which were all the byproducts derived from mussel, from Pilus galliprovincialis, and were cells, broth, broken viands and breeds, along with the epibiome. And the third group would be the cephalopods. That wasn't all the byproducts that we work with. These derived from, let's say, this group, were from a mix of 4 species of squid, and it comprised viscera, glavius skin, and cooking broth. And lastly, within this group, we have the cooking broth as well of Octopus vulgaris. Next slide, please. Okay, and finally, we also work with 6 species of macroalgae from different groups as well, let's say, that were Ulva rigida, Porphyra umbilicalis, Cystoseira usneoides, Saccharina latissima, Rugulopteris okamurai, and Gelidium cornum. Next slide, please. I will give you an overview based on each group of samples to try and facilitate a little bit the results that we got. So first we did proximate nutritional composition, and we found that protein was the major component in fish and cephalopod byproducts, whereas as was the principal component in mussel due to the inorganic nature. However, in macroalgae, what we found most common was the carbohydrate content up to 60%. Next slide, please. Okay, if we have a closer look to the lipid content, that of skipjack tuna heads and turbot viscera ascended to 38 and 50%. Some similarities were found as well across these these different groups, like the high content in saturated fatty acids and the abundance of palmitic and stearic acids across the samples. Regarding monosaturated fatty acids, fish byproducts contain a higher percentage than muscle. But again, it's worth mentioning skipjack, tuna heads, and turbo viscera due to the remarkable content. Lastly, polyunsaturated fatty acids were present in lower concentrations. Still, EPA and DHA were detected in all samples of both groups. However, the concentration of DHA in skipjack, tuna heads, skin, and viscera was remarkable. So it's important as well to take this information into consideration for the fermentation application and the next steps in the project. Next slide, please. Okay, regarding cephalopods and macroalgae, the saturated fatty acids comprise most of the fatty acids present in the samples, with again stearic and palmitic acid as the most abundant ones. Likewise, monosaturated fatty acids were comparable between both groups. However, it was observed that the concentration of polyunsaturated fatty acid in squid skin and viscera reached up to 50%. And DHA was detected in both groups, so EPA was not present in cephalopod byproducts. Next slide, please. Okay, so now we move on to the amino acid profile that was not originally contemplated in the project. We measure both the essential amino acid and non-essential amino acid. Even though it's a little bit higher in fish byproducts, it's in range, it's overlapping with the mollusk bivalves byproducts. However, leucine and lysine in both groups of samples comprise the most abundant amino acid, followed by isoleucine in fish byproducts. And in the case of viscera from skipjack, tuna, and yellowfin, the concentration was especially higher, which is in line with as well the higher protein content of these samples. About non-essential amino acid, they comprise most of the sample, with glutamic acid, and glycine as the most abundant ones. Next slide, please. Regarding cephalopods and byproducts and macroalgae, the content of essential and non-essential amino acids was quite similar. However, some differences can be spotted. Like for instance, in squid and octopus byproducts, as well, again, leucine and lysine were the predominant essential amino acids, whereas it should be highlighted that squid viscera was rich specifically in isoleucine and squid glavius in histidine. On the contrary, about macroalgae, it should be noted that phenylalanine was present was the amino acid with the higher concentration in Gelidium corneum, which is important since this is the base that we typically use to as well synthesize adrenaline and its analogs. Finally, about non-essential amino acids, in the case of cephalopod byproducts, glutamic acid and glycine, as in the previous groups, were the most abundant. However, in macroalgae, it was aspartic acid instead of glycine. Next slide, please. Okay. To finalize with the nutritional characterization, I'm going to talk about a little bit the results that we got for the mineral composition. And here we analyze some macroelements and some microminerals. Sorry. In the case of fish byproducts, as well as in the case of mollusk bivalves, calcium was the most abundant or most commonly found across throughout all the samples, and this is in accordance with the fact that in fish byproducts are some we can find some bones or have high content of them, and as well due to the inorganic nature of most of the mollusk bivalves byproducts. In the case of fish, sodium was the next in line about the mineral composition. However, it should be taken into consideration that tuna cooking broth and they comprise up to 11 grams per 100 grams of sample of sodium, which is quite high. On the other hand, in mollusk bivalves, even though calcium and sodium were the most abundant, cooking broth was enriched in magnesium and potassium, which should be, or we had taken as well into consideration to select these samples to fermentation. Finally, regarding microelements, or iron and zinc were again the most abundant. In the case of fish byproducts, skipjack, tuna, viscera were the one comprising the higher concentrations, whereas in the case of mollusk bivalves, mussel epibiome was a considerable or a source of magnesium, which should be taken into consideration. Next slide, please. Okay, so regarding mineral composition of cephalopods byproducts and macroalgae, the case of squid and octopus byproducts, we found that calcium was not that abundant. Instead, it was sodium and potassium. And when talking about macroalgae, calcium was, and potassium as well, the most abundant. However, sodium and magnesium were not that different from the concentration that we found of calcium and potassium, which poses macroalgae as a balanced composition of micromineral source. About microminerals, zinc and copper were the most common ones in squid byproducts, with gladius being the highest in zinc content and viscera about copper. When we assess the mineral content in macroalgae, especially about the microminerals, even though iron was the most abundant, again, iridium, cornium comprised a quite balanced source for iron, magnesium, and zinc. And a very interesting difference that we found from macroalgae when compared to the other three groups is that nickel and cobalt were detected. Next slide, please. Thank you. Moving on to the food safety assessment, we perform cell viability assays in 2 cell line models, one coming from liver and the other one as a model of intestinal barrier. We carried out 2 different methods based on cell metabolism. And as a result, we found that across the three groups, fish, mussels, and cephalopods byproducts, the concentration that we tested didn't reduce cell viability. However, these 3 macroalgae reduce cell viability at high concentrations. So this should be taken into consideration when we move forward to further steps in the project. Next slide, please. Due to the nature, again, of these samples, we checked the marine biotoxins presence. These toxins can be classified in 3 groups, lipophilic, amnesic, and paralytic shellfish toxins. They are regulated. And even though we didn't detect any amnesic or paralytic shellfish toxins, we did for the lipophilic ones in squid viscera and mussel broken biomes. However, even though they were present, they were still in the within safety regulation, within the legal limits set by the legislation. When we assessed as well the heavy metals, we tested lead, mercury, arsenic, and cadmium. And even though very low concentrations of these metals, the concentration of arsenic was especially concerning in Phystoceira usneoides, which can explain the decrease in cell viability when we tested the extract. So these 3 macroalgae should be taken into consideration, this information for the following steps. Next slide, please. Okay, so based on the nutritional characterization and the safety data, the marine byproducts that were selected to optimize solid state fermentation and liquid state fermentation were those derived from tuna, head, skin, tails, central spine, cooking broth, and viscera, due to their availability and protein content. From the mussel group, epibiont, broth, and broken biont substrates were also chosen based on their especially their mineral composition, and ulva rigida and Delidium corneum were as well selected for these next steps to provide the carbohydrate content. Next slide, please. Okay, so based on all of these, I have talked to you about the nutritional characterization and food safety of all of these quite diverse biomasses. And this entails the work package 2, which is finished already. And ongoing is the byproducts fermentation to both are ongoing right now by 2 of our partners to generate and provide with the proteic process sample, from which we will continue with the next steps. Next slide, please. Right. And to conclude this presentation, just as take-home messages, we saw that marine byproducts were rich in protein and inorganic material, which constitutes a good base to perform fermentation and obtain the protein extras. that the presence of heavy metals reduce their viability, discourages the use of Schistocereus neoides and Porphyra umbilicalis. And this closes with the selection of tuna and mussel along with Ulva rigida and Delidium corneum for the fermentation steps. Next slide. Thank you very much.
Thanks a lot. So here you have the contact for PROMISEANG. And amazing. So we are done with the presentations. We are perfectly on time, and we are still 40 participants, which is a great number. And this shows your interest for these research activities and results. And now it's time for the Q&A session. I first of all invite you, if you like to present yourself on the chat, so maybe other participants can reach out to you or find any synergy or opportunity between your organizations. For example, in the participants here, we have representatives from different sectors. Mainly we have academia, but also we have research centers, NGOs, technicians of the fish and animal sector, consultancies, but also policymakers. And I saw also a risk assessor for food. So we have a really varied audience today. And I invite you to share if any question or any even the goal that of why you joined today's webinar. If you were looking for some connections or you were just curious to get to know our project. Okay, so Patricio, nice to meet you. I'm happy that you used the chat to introduce yourself. I think no, it feels like there are no questions. So maybe it was even a lot to digest. We've been here for almost 2 hours, and with the dense information. I remind you that we will be sharing the presentation of today's webinar. Also, it's recorded, so you will see it on the one of the project's YouTube channel. We will inform you via email. And then I can introduce to you that we have other thematic webinars upcoming in the next year. For example, One Earth is almost 1.5 years old, so we're not even half of the project, and now we are starting to see the first result of the research. I see that you are sharing, I think, your organizations or some articles, which is really interesting. Yes, so what are your preferences? Shall we leave the room open? I see that people are more comfortable in sharing via chat information or themselves. So I think it's good to leave it open and use also the text format for communicating.
I think there's a question here, Leticia.
Okay, sorry, didn't see it. Yes.
From Teresa Cesario from the PROMISEANG project, would like to know in the Valorish project if anaerobic or aerobic fermentations are done for the proteolytic fermentation. So maybe one of our experts here, our partners.
Yeah, can you hear me?
Maybe Borja, yes. Okay, thanks, Borja.
Okay, you're welcome. We are using right now aerobic bacteria, but we are planning to use also anaerobic, but anaerobic facultative bacteria as well.
Guys, I don't know if you have any other comments. No. Okay, so let's go to Gabriela Borrell. She says that she's a student at UAM and did her master thesis on PROMISEANG project last year. So she was curious